Students, welcome to the third video of your biology classes. We were discussing about types of solutions in osmosis. So there are different types of solutions: hypotonic solution, hypotonic solution, about which we have already discussed. Second one is hypertonic solution, hypertonic solution, hypertonic solution, and all these we have already discussed. Hypotonic solution means a solution in which lesser amount of solute in it, and a hypertonic solution is a solution in which more amount of solute in it comparing to the solvent. And there is one more type of solution which is isotonic solution. Isotonic solution. Isotonic solution contain equal amount of solute and solvent. The percentage of solute and solvent in isotonic solution is almost equal. So these are the three types of solutions. In hypotonic solution, the amount of solute, for example, if we are making a salt solution, salt is in a salt solution, adding common salt in water, that is salt solution. We can make salt solution by adding common salt, table salt in water. If in this salt solution the amount of salt is more, अगर वो गाढ़ा होगा, the amount of solute is more, then it is a hypertonic solution. If there is less salt and more water, then it is a hypotonic solution. If there is less salt and more water, then it is a hypotonic salt solution. If there is more salt and less water, then it is a hypertonic salt solution. It is isotonic when the amount of solute and water are equal, then it is isotonic solution. Now when we put a cell, a cut part of any plant part, you cut and put in this solution, in these three different types of solution, then what will happen to it? You cannot add cell into it. Now you can practically add the parts of living organisms in it for an experiment. Suppose you are adding raisin, kishmish to all the three type of solutions. You kept hypotonic solution in a beaker, in a glass, or and another glass vessel, tumbler, you have taken hypertonic solution, another glass you have taken isotonic solution, and now you are putting raisins in all the three solutions. Then after some hours or the another day or overnight, when you observe those raisins, you can see the change in raisins. How can you see the changes? In a hypertonic solution, the solute is less and solvent is more. So you can see the raisins are swelled up because the amount of water is more, so the water is absorbed by the raisins. Amount of water is more in the outside medium, in the raisins, inside the raisins the amount of water was less comparing to the outer surrounding. So in a beaker you have the solution, or in a glass you have the solution here. You have the solution here in a glass and you put some raisins in it. And these raisins absorb the water from the surrounding. In the resins there are cells, and those cells absorb water from the surrounding. So absorb water from the surrounding because it is a hypotonic solution. Because it is a hypotonic solution. In hypotonic solution, the amount of solute is less and water is more. So the resins are swelled up. Resins swelled up. And now if you put some resins in the hypertonic solution, you cannot see the resins swelled up because the solute is more there, water is less. So hypertonic solution example, concentrated sugar syrup or concentrated salt solution. These are all example of hypertonic solution. So in those hypertonic solution, the resins will not swell up because like in a hypotonic solution, because there is less content of water, more content of solute. In isotonic solution, equal amount of salt and or sugar or any solution we take, equal number of solute and solvent are there. So there will be no change for a cell. If a normal cell, we are putting a normal cell in an isotonic solution, the cell will not be swelled up or it will not be shrink, because the cell will shrink in a hypertonic solution. There will be no change for the cell in an isotonic solution. The cell will be swelled up in a hypotonic solution. So in a hypotonic solution, the cell will swelled up. Swelling will take place for this because by absorbing water. Hypertonic solution, it will be shrinking because losing water. Lose water to the surrounding because in the solution there is less water, so the cell will lose water. Now, so this is swelling and shrinking is take place by osmosis. So the process took place here, the process taking place here is osmosis. So water is absorbed by osmosis in hypotonic solution, so the cell is swelled up, and hypertonic solution the water is lost by the cell, then it is shrinkage of cell take place. In an isotonic solution, no change take place, no change. There will be no swelling, no shrinking. Isotonic solution, there will be no change. So these are the three types of solutions. This is an important topic, osmosis and the types of solution. These are important topics in your chapter 5. Another thing is, in hypotonic solution and hypertonic solution, what happened? If water is taken in, when water is taken in and the cell is swelled up, there take place endosmosis. Endosmosis take place. So endosmosis. Endo-osmosis. It is endo-osmosis. End-osmosis or endo-osmosis is because of